So hello once again. I'm by name Bahore Ahmed. I'm the student at Ines Ruhengeri Year 3 and my reg number is 23 slash 19560. So I'm going to present about the questions from question 14, from question 11 to question 15. So the first question is, what are the risks of chimeric antigen receptor T cell therapy? So this CAR T cell therapy involves in engineering a patient's T cells to target the specific antigens, typically on the cancer cells. The risk of this CAR T cell therapy may include the following. It may include, firstly, the cytokine release syndrome. This is a severe systemic inflammatory response caused by a massive cytokine release. Its symptoms, it may include the increase in temperature, which will result in fever, hypotension, and organ dysfunction. The other risks that include its neurotoxicity manifest as confusion, seizure, or cerebral edema, also known as immune effector cell associated neurotoxic syndrome. The other risks that include CAR T cell therapy is on target of tumor effects. CAR T cells may attack the normal tissues expressing the target antigen. And the other risk that include is infections, tumor lysis syndrome, and graft versus host disease. On the infections, immunosuppression of loss of B cells. This is mainly from the CD19 target therapies, and it increases the risk of infection. And on the TLS is the rapid destruction of tumor cells leads to the metabolic disturbance. And lastly, on the graft versus host disease, through the rare of CAR T therapy, it may occur if the donor T cells are used. And the following question is, how is immunotherapy used to treat autoimmune disease? In immunotherapy, autoimmune disease aims to modulate the immune system to restore the tolerance to the self-antigens. The approaches may include monoclonal antibodies. This will target the specific immune components to reduce the inflammation and autoimmune. It will also target the anti-TNF. Example, this here an example we may include the infliximab. It is used in rheumatoid arthritis and Crohn's disease. And it will we may also get the anti-interleukin 6 that reduces the inflammation and giant cell arthritis, and also the anti-CD20 that duplicates the B cells in the systemic lupus erythematosus and the multiple sclerosis. We can also talk about the immune checkpoint inhibitors. This is the block of the costimulatory molecules to suppress the overreactive immune response. We may also talk about the intravenous immunoglobulins. This will provide the immunomodulatory effect in the disease like as Guillain-Barré syndrome and myasthenia gravis. Cytokine targeting agent, we have anti-interleukin 17 and interleukin 23. We also have the anti-IFN, which will be used in reduction of lupus activity. The cellular therapies also will regulate the T cells of the stem cells or being exposed to the restore tolerance. The third question which I'll be talking about is, how do T helper cells support the B cells activation? T helper cells, particularly T follicular helper cells, play a critical role in B cell activation and function through the direct interaction of cytokine secretion. The first point will be the antigen presentations. The B cells internalize antigen bound to their B cell receptors, process them, and present the peptide fragments on MHC class II molecules to the T helper cells. The T helper cells also recognize these antigens via their T cells receptors, which can be called TCR. We will also have the costimulatory signal CD4 and CD40 and CD40 interaction on T helper cells bind the CD40 on B cells, a key signal for B cell activation and survival. Cytokine secretion will also help in the activation. The T helper cells release cytokines. This means interleukin 4 and interleukin 21, which will promote the B cell proliferation. This will be the manufacture of B cells. It will also help in enhance antibody production, facilitate germinal center formation. Through these mechanisms, all these mechanisms we talked about, the T helper cells drive B cell differentiation into plasma cells and memory cells. For the fourth question, we'll talk about what is the isotope switching and how is it regulated. For the definition, isotope switching, it may also be called or also known as class switch recombinant. It is a process where the B cells change the class of antibody they produce. This means that it may change IgM to IgG or IgA or IgE without altering the antigen specificity. The key features here we can say about this isotope switching. We can talk about what's the purpose of isotope switching. To tailor and the main purpose is to tailor antibody function to specific immune response. This means that, for example, you can say about when IgG for systematic immunity and IgA for mucosal immunity. And the mechanism of this isotope switching is the constant region C. This is the main part that found on the antibody on the heavy chain is replaced guided by switch on S regions of the immunoglobulin gene. Requires the activation induced cytogen diamine aid to induce the DNA breaks into the S region. And the other point we can talk about is for the regulation, the cytokines secreted by the T helper cells interleukin 4 induces the switch to the IgE or Ig1. We may also talk about the CD40 to CD40L interaction. This is the essential for the aid that we talked in the previous slide activation and CSR initiation. Defective isotope switching can lead to the immune deficiency such as the hyper IgM syndrome. And the last question I will be presenting, it will be what are the T follicular helper cells and why are they important? These T follicular helper cells are the specialized subset of CD4 plus T cells that reside in the lymphoid follicles and are essential for the adaptive immunity. So we can talk about the functions of these T follicular helper cells. The first function is B cell help. T follicular helper cells provide the signals that promote the B cells proliferation, differentiation, and survival. It will also help in the germinal center formation. They support the formation of the germinal centers where the B cells undergo the somatic hypermutation and affinity maturation to improve the antibody specificity. We can also present about the isotope switching, where the cells secrete cytokines like interleukin 4, interleukin 21, and IFN, which directs to the isotope switching of the B cells. The memory B cells generation. T cells help to generate the long-lived memory B cells for the sustainable immunity. What lastly we can talk about is the importance of these cells. Without these cells, germinal centers fail to develop, leading to the impaired antibodies response. This deficiency underlies the condition such as immune dysregulation syndromes or suboptimal response to vaccines. And this was my last presentation. Thank you for your kind attention.